we're going to look at vascular transport in plants, and specifically for this video you need to keep in mind several things. First up, how does the initial absorption of water happen? So how does that get into the root? The next big question is looking at how does the water get into the xylem, and then finally, how does the water get up the leaf? So we're going to go through each of those in turn. Right, so first up we're looking at the types of kind of transport systems that you may see within the plant. And so it's a typical plant where you can see the roots down the bottom here, and then you can see basically you're going to get vessels that run up the plant through the stem, and then up to the different sections on the leaf, and also up to the flowering parts of the plant. Now, in terms of the actual structures involved here, we're going to be talking about the xylem, and the xylem are a set of tubes which effectively will carry things like water and dissolved minerals and inorganic ions from that are absorbed from the soil up through the xylem and then up to the leaf where they're required. So in terms of some of the ions that you might need to be aware of, nitrates would be one, magnesium, potassium maybe another, phosphates maybe another, and those are the types of things absorbed in the roots that then can get into the xylem. So something else that we'll look at in maybe later videos, which is looking at the structure and the kind of function of the phloem, which carries organic materials, so in this case sucrose and glucose. So just as a bit of a recap for the different types of transport that occur across membranes, you've got diffusion, osmosis and facilitated diffusion. These are all passive types of transport. The key differences here: osmosis needs a protein and it transports specifically water. Facilitated diffusion is diffusion with a protein in effect. And the reason why they're passive and they don't require ATP is because it's down the gradient. Active transport does require ATP, and therefore it's going against the gradient, and it also requires a protein. Hopefully something that you should remember as well from looking at water potential in some of the previous kind of sections, then effectively the water potential is determined by a couple of things. It's determined by the solute potential and therefore the pressure potential. So those two things, in simple terms, if it's got more solute in it, it's going to be more negative. And so all water potentials are addressed as negative unless it's completely pure water. Then it has a water potential of 0, and the more solutes, the more dissolved ions that you have found within the solution means it's going to be more negative, and effectively the water will move from an area that's less negative to more negative water potential. So how is this relevant? So the first major big question is why the water gets into the root hair cell in the first place. So if we imagine that this is our root hair cell, we imagine that this is the outside of the root, so this is the soil, then what's going to be happening initially at that root hair cell is active transport is going to be happening, and basically what's going to occur is these inorganic ions, these potassium ions, the types of ions like nitrates and magnesium getting pumped into the cell. And what they're going to do is, because they can obviously dissolve in the water, they're going to reduce the water potential and make it more negative. And by making it more negative, that's going to then increase the rate of osmosis into the cell. So that's going to physically drag more water in across the membrane. And you can see that kind of illustrated here. So in a high solute concentration, so if it's got a lot of inorganic ions, like it has inside the cell, those inorganic ions are dissolved in the water. Therefore, the water is going to move to the more negative water potential, so they move across the membrane and into there. And so that's how the water gets in initially. Okay. In terms of the second big question, so this is how the water then gets into the xylem. So if you look at the overstructure of a root, what you've effectively got, as you can see it here, this would be our root hair cell, and then from here you've got a series of other cells in the cortex, and then in this central section, kind of in the root, you have the xylem, and then the xylem can then run, if you like, laterally, going all the way up the plant. So the question is, how does it get from here into there? So what's effectively happening, there are two major pathways between these cells, and the first major pathway is referred to as a symplastic route. Now, one thing you may not be aware of with plant cells is that they have these kind of channels and these holes between the cytoplasm of different cells, and we call those holes plasmodesmata, and so it means that the cytoplasm from this cell is joined with this one, and so in simple terms it means that anything in the cytoplasm of this cell could move directly through the cytoplasm of other cells, and so we call this the symplastic route. The other route for the water to move through is along this apoplastic way, so between the gap between the membrane and the cell wall, and so effectively the water can run through here. Now, something I'm not entirely sure would be necessarily on our spec is the presence of this, which is the Casparian strip in the endodermal layer. So this Casparian strip basically runs across the gap between the cell wall and the membrane in the apoplastic route. So what this basically means is all of the water that gets into the xylem initially has to travel through the symplastic route. It can go apoplastic route all the way through here until it gets to here, and then it needs to go into the cytoplasm and therefore into the xylem vessels. And so this is just an overview of effectively the same thing. So symplastic route goes through the plasmodesmata, and so this can just happen through simple diffusion, so the water will move from an area high concentration to low through each cell, and in this case it could also move through the apoplastic route between the cell walls and the membranes. Okay, so the next big question is how the water gets up the leaf, and so in this case the water moving up the leaf, there's a few explanations for this. One of the first things is capillarity. So you may have seen this with tubes, or you may have seen this with straws in water. So if we've got a water source here and we dip different width tubes into the water, you'll find that the water actually rises up these tubes higher than the level in the, let's say, the beaker that it's been placed in. And this is down to capillarity. And the reason for this is the water molecules adhere to the walls of, let's say, the straw in this case. Now, if you consider that in a thinner tube, then you're going to have more water molecules adhering to the walls of this, because in effect, if you look at this middle section here would have very few or no water molecules adhering to the walls. And if you look at the middle section here, the middle section is much larger, and so a higher volume of the water molecules won't be adhering to the walls, and therefore it doesn't rise up as much. The real consequence for this is the fact that the tubes are very, very thin, get the water to rise up the most. And so this leads to the idea that the xylem are incredibly thin, and that helps with this capillarity. The problem with this idea is the fact that capillarity only really explains how it can move up maybe a couple of meters up the plant, so not very much. Another explanation for how the water could potentially move up the plant is root pressure. So in this case, because there's lots of water moving into the roots, then effectively that water will exert a force, and that force is referred to as root pressure, forcing the water up these narrow tubes. Again, the issue with this is this doesn't fully explain how the water moves up extremely large plants. So the final explanation is the cohesion-tension theory, and we'll come to that in a sec. But it's probably worth having a good look, really, at some of the structures involved in the xylem cells. So with the xylem, they tend to be a continuous tube of dead cells. So effectively they used to be living cells that then died, and in effect the end walls between the different cells have gone, and so it just forms one long continuous tube of cellulose cell wall. Some of the things that you should probably be aware of is that the xylem carries the water up the plant in only a single direction. The walls are dead, and so these are dead cells. So this is effectively the cellulose cell wall that's left behind. These walls have lignin in there, and this lignin normally forms kind of circular, kind of spherical arrangements that you would find in here. So you may do a practical with beetroot where you can actually see this. And so these lignin rings that you get actually add waterproofing, and they also increase the strength of these vessels. And this enables the water to be able to move up via cohesion and adhesion. Adhesion means it's adhering to the walls of the xylem, and cohesion means that it's going to join onto one another. So cohesion is bonding between water molecules. Adhesion is where the water molecules bond to the walls. So the final explanation for how the water moves up the plant is referred to as the cohesion-tension theory. So in this case, how this works is this represents, so this diagram here represents the water molecules moving up the xylem. Now we know that water is dipolar, which means it has a positive end and a negative end. This means that the positive hydrogen end of the water will form hydrogen bonds with the oxygen, so the negatively charged oxygen. And this will mean that you'll basically get a long string of water with these hydrogen bonds. Now the consequence for this is when the plant transpires, and transpiration is known as the diffusion and evaporation of water away from the plant, when one of those water molecules diffuses out, that's going to reduce the water potential inside the kind of spongy mesophyll that you get in here. And because these water molecules have gone through, in this case, cohesion, because they're all stuck together, when one evaporates out, then that string that's all joined due to cohesion is being able to be pulled up the xylem tube, and therefore the water can be moved up the plant to enable it to do photosynthesis in the cells in the leaves. And so this theory is probably the best explanation we currently have for how the water moves up the plant. So this diagram should hopefully start to show some of the differences here. So in this case, what we've got is the water evaporating away at the leaf, and then because the water molecules have gone through, in this case, cohesion, they form hydrogen bonds between one another. When one is pulled up and evaporates away, it will effectively pull the rest up in tension. So if you remember, it's called cohesion-tension theory, and therefore this means it's going to pull the water up from the root in this kind of long string that you get here. The other thing that really helps here is the fact that you get adherence as well. So when these water molecules are moved up, they're going to adhere to the walls, and you get this, basically this pulling of the water molecules in tension, and that's how effectively the water gets up to the top of the plant. One of the things that's probably worth kind of mentioning is the idea of being able to measure transpiration, and one of the ways in which we do this is we use a piece of equipment called a potometer. So in a potometer, what we would do is we would take our plant and we would set it up so that we'd fill all of this reservoir and all of these tubes with water, and effectively we would place an air bubble here in this tube, and what you could basically see is you could see the rate of transpiration by seeing how quickly this bubble moves along here. And if you know the width of the tube, you can basically work out, in this case, the volume of water transpiring away from the leaf. So let's say you were going to look at, I don't know, the effect of light intensity on the rate of transpiration. You'd put your light next to the plant, effectively see how quickly that air bubble is moving, so to work out the rate of transpiration, alter the light intensity, and therefore you could work out how quickly the plant is transpiring, and you could see what effect that could have. This practical could be done for not just light, could be done for CO2 levels, could be done for humidity, could be done for a number of different factors that we know affect the rate of transpiration. In terms of the types of things that actively change it, it would be light would be one, would be an obvious one. Temperature has a massive effect. Also, humidity has a massive effect on this, on the plant's rate. But that's definitely worth considering looking at, and it's a type of question that they might commonly ask.